Hello everyone, welcome to Dr. K. PREM-PRIMER Biotech lectures presenting by Dr. Prem. Today's lecture is talking about biotechnology briefly and what are the applications of biotechnology. Everywhere you can see this is the application of biotechnology. This is the application of biotechnology. Everybody is attributing a lot of things to the biotechnology. Hence, I would like to present what is. Let us define the biotechnology, right? So then, define biotechnology and its applications of biotechnology. We will see today in today's lecture. As you all know that the term biotechnology is coined and introduced by Carl Erke. So he is a man. Carl Erke is the founding father of biotechnology. He is the founding father of biotechnology. So he was a Hungarian agricultural engineer who wrote a book about biotechnology in 1970. It got published in the 1919, where he coined the term biotechnology. Hence, he considered as founding father of biotechnology. Founding father of biotechnology. You can see, founding father of biotechnology is none other than Carl Erke. In 1917, he wrote a book. In that book, he introduced the word called as biotechnology, and also he defined what is biotechnology. If you ask what is biotechnology in terms of Carl Erke, you see that his definition is biotechnology was simply the process of using technology to convert the raw biological material into a useful product. He said that the biological materials or biological things which are converted into the useful product whenever you use a technology. Technology can convert the raw biological material into a useful product. That is called as biotechnology in view of Carl Erke. He wrote a book that is Biotechnology of Meat, Fat and Milk Production in an Agriculture Large Scale Farm in 1970. In that book, he coined and introduced the word biotechnology. So, biotechnology was simply the process of using technology to convert the biological raw material into a useful product. So technology is converting the biological raw material into a useful product. That technology or that process is biotechnology. Right? So I would like to explain biotechnology with this cartoon. You can see this is the corn. From corn, you can get what you can get. You can get grains, corn grains, and the leftover is corn stover. Corn stover. This is the corn stover, right? This is the corn stover. So corn stover, or agricultural residue is used as a animal feed. Cows, buffaloes, or other animals can use this this agricultural residue as a food. But grains can be used. Grains can be used for different purpose. Like you can see, the grains or maize grains can be used as a staple food, right? Staple food. staple food, and industrial meats. It can be used as a industrial purpose, and most of the maize grains are used as a feed for poultry and other livestock. You see, the major percentage of production, the maize grains are used for feeding of animals and birds, right? But this is all known. But biotechnology is all about using of the process of technology used to convert the biological raw material into a useful product. Now you can imagine that anything, if you are using the maize grain for feeding of animals and birds, and the agricultural residue is for the feeding of cows and buffaloes. This is what we can imagine, anybody can imagine. But beyond that, you can expect a bioethanol from the corn stover, or as well as the same bioethanol from the maize. Can you imagine? That can be imagined only when you use the biotechnology. See, maize utilization. Bioethanol production in US, in that area, the United States is leading. You see, United States is leading. Around 16,061 million gallons of bioethanol is producing by using the maize grains. And the next one is Brazil. You can see 7,920 million gallons of bioethanol is produced by using the maize grains. So this is what a very simple one, but you see biotechnology was simply the process of using technology to convert the raw biological material into a useful product. That's a simple one. Today also the same definition is almost eligible for every biotechnology aspect. Nothing much different from that. You see, here I want to tell you here, this is a corn store. When you, corn store is used as you all know that what is happening in the Haryana and Himachal Pradesh, where the agricultural residue is burned, that's becoming a big problem. But here, the corn store can be used as a feed, you can use for the feed. But here, the corn store is milling and making a small chopped into the small, and then it is treated with the acids, and once it is ready for the saccharification. Here, what you are doing is you identify a microorganism which can identify a microorganism which can digest the lignocellulose. So you all know that the plant cell wall is consists of lignocellulose. Lignocellulose means lignin, cellulose, hemicellulose, and pectin. So most of the cell wall is composed of this, and less amount of protein and other fats. But 80 to 90% of the cell wall is all about carbohydrate. So the carbohydrate that's in the form of cellulose and hemicellulose. If you can identify a potential microbe which can convert the hemicellulose into the xylon, other pentoses, or cellulose into the glucose molecule. So once you identify a potential microbe, so if you do the saccharification with that microbe, then what will happen? The lignocellulose or cellulose becomes the glucose. That glucose can be undergoes the fermentation with the one more usage of one more microbe, particularly yeast. Then glucose is converted into the ethanol. Then that ethanol is distillated and you can use for the running of any vehicles. So this is what we can't expect, without biotechnology. Similarly, if you see corn majorly it is used for the feedstock. But whatever leftover can be used also to get the biofuels from the age. You can see corn and milling and saccharification, then fermentation, and you get biodiesel or bioethanol from it, right? So trying to tell you that by using the technology, we can convert the biological raw material into a useful product. That is called as a biotechnology. You know, we never imagined that we can get a bioethanol from the corn stover or corn grains. This is possible with the biotechnology. Yeah. So also there are many versions of this biotechnology. You can see the different definitions. Exploiting the properties of biological material with the technology to provide a useful product. Exploiting the properties. What property? Each and every biological material have a different property. So the property can be neutralized or increased or decreased or modified. So with the help of technology, then that becomes a very valuable product. So that's what we can say, exploiting the properties of biological material with the technology to provide a useful product. That's what is biotechnology. And you see here, as at its simplest, biotechnology is technology based on biology, biotechnology. Biology, biotechnology, harness the cellular and biomolecular process. You all know that whenever we are tired, it means that, you know, our respiration, there are two types of respiration. One is oxygen based and other one is anaerobic, where oxygen is not there, right? So when you have enough oxygen, then glucose converted to the pyruvate, and pyruvate goes to the mitochondria. There it goes the electron transport chain. It goes to citric acid cycle, produce NADPH and FADH. Those NADPH and FADH can convert to the ATP through the electron transport wave. That's in the presence of enough oxygen. When you don't have oxygen, the same glucose molecule converted to the ethanol, right? Here I am trying to tell you all that we are trying to replicate the biological process in vitro condition. Then you get a better products, right? Whatever is happening within the cell, same thing we are trying to replicate in a laboratory condition, right? So that's what you can get and you see the technology and products that help to improve our lives and health of planet. You all know that you have insulin or erythropoietin or interferons or antibodies. They can be produced in laboratory, right, with the help of technology, so that we can increase the health of humans as well as the planet Earth also. We can create a superbugs which are having a ability to degrade the pollutants which are the byproducts of industrialization, right? So to save the health of both the human and the planet, we can use the biomolecular process happening in the cell. There is another term. We can explain the same biotechnology in a different way also. Technologies that make use of living organisms or its biological molecules to generate a useful products for the benefit of mankind. What we are trying to tell, we can use the living organisms or its biological molecules, right? Technologies that make use of whole cell or its macromolecules to generate a useful products for the benefit of mankind. So I am trying to tell you that technologies can be used to make use of living organisms. For example, if you take lactobacillus, so you just add lactobacillus to the milk, that will convert to the yogurt or curd. So where you are taking entire cell or entire living organism and converting the milk to the yogurt or curd. Or you take a juice of any fruit juice converted to ethanol, where you are using the yeast. Or you can take biological molecules like enzymes. Directly you isolate enzyme from that organism, and that enzyme can convert the raw material into the product. That is also called as biotechnology. Here I am trying to tell you that biotechnology is not an independent entity. It's a multidisciplinary subject. I can show you the same thing here. You see, cell and molecular biology. Cell biology means where there is a relationship between the structure and function. If you take a human cell, or you can take the prokaryote or eukaryote cell. Why mitochondria is like this? Because it has to perform it produces the energy. To produce the energy, there is a certain requirements in the structure, right? So cell biology or molecular biology, how transcription is happening in the eukaryotes or prokaryotes, or replication, means DNA to DNA or RNA to RNA, what are the enzymes are involved in that one, right? Cell and molecular biology. And the microbiology is all about study of the microbes, right? So what is the structure of microbes? How many classes of microbes are there? What are the use? How they look and what are the pathogenicity? This is all about microbiology. And genetics. Genetics means DNA, genes, right? Exons, introns, transcription, translation. It's all about genetics. Biochemistry. All nucleic acids, carbohydrates, proteins, how they are made of, right? Physiology. I'm trying to tell you all that in a brief. Phenotypic, phenotype is all depends on the genotype. So genotype to the phenotype, we can study the cells, prokaryotic cells or eukaryotic cell with the cell and molecular biology or microbiology, genetics or biochemistry. So whenever you study these aspects with the prokaryotes and eukaryotes, so when you identify certain traits, those traits can be processed to you can make them in a bigger by using the technology called RDN technology. For example, you identified a gene, right? You want to have more copies, then you have to clone through the RDN technology. For example, you identified a antibody which works against a certain pathogen, then you develop such a certain antibody through the immunotechnology. For example, you identified a microbe which can produce the more enzyme, right? Certain enzymes. Then what you have to do is you have to create, you have to develop a fermenter where you have to give the raw material by using the cheap and most available raw materials. By using that, you can produce the enzymes, or you can produce any other product. This is called as a bioprocess technology. And here you can bioinformatics. Whenever you say the enzyme is inhibited by some natural products, right? So then you can search these similar products, or you can change the certain moieties of that natural products or you prepared products. By using the bioinformatics, you can develop a drug which is having more potential, right? And tissue culture, here you can, suppose you identified a plant which is having high medicinal value. But if you go through the normal culturing, it takes years together to get 1 million of plants. But if you use the tissue culture, right, where you take the small amount of explant from your desired plant, and convert it to the callus, and callus again distributed to the various one, and you get millions of same plant within within a room or within a house within no time. That can happen with the tissue culture. So whatever we have identified properties of cells through the cell molecular biology or microbiology and genetics, and they can be used, these technologies to produce, microbial biotechnology to produce, microbial technology medical biotechnology, animal biotechnology, and plant and environmental biotechnology. Here, when you use the microbial biotechnology, you can produce the antibiotics and yogurt. I'm just giving two examples. And if you say medical biotechnology, you can gene therapy, vaccines, and diagnosis of disease. So, if I have a particular mutation in my genome, when you identify that mutation in my genome, I am able to know that whether I am prone to particular disease or not, because that can be known through the diagnosis. Means you can use your genes and the mutations to tell whether you are going to develop that particular disease or not. That can be, you can predict your future disease also. And animal biotechnology, where you can you see monoclonal antibodies or monoclonal antibodies and bioinformatics, and animal models for you can develop the animal models for human diseases. So certain diseases are not there in the they are not there in the animals. So when you change the genome, and they also can develop the certain disease model, so that you can study the pathophysiology as well as the treatment using those animals. That can be done with the animal biotechnology. And plant and environmental biotechnology, where you all know very famous example, BT cotton, where cryogens of Bacillus thuringiensis were isolated and inserted into the cotton, and that is now it is resistant to the insects, right? And bioremediation, if you identify genes, those genes are going to degrade some aromatic compounds or genobiotics. You can insert those genes into the vectors and transform into the E. coli or other hosts. Those hosts can clean that contaminated or polluted lands. And biopesticides. So you can develop the biopesticides also. So this is what is biotechnology. Biotechnology is not a single entity. It's a combination. It's a multidisciplinary, right? So you should have a knowledge of all these subjects, then only you can come up with some products. So when you have the knowledge of all these subjects, right? So those knowledge can be used to develop technology. From the technology, you can convert the raw material into the valuable product. I'll give you a few examples, like you see. You see, here I am saying living organisms. Biotechnology can use the complete organism, or it can use a macromolecule, or it can use the certain secondary metabolites. Right? It's not that it is not that only only whole organism or DNA, RNA or not. It's not like that. Depends on the property. You can make use of whole organism. Like you can see here, living organism, fully active. Fermentation of curd, you can see lactobacillus. Alcohol production, you use the entire organism. To produce a curd or alcohol, you use the yeast, right? Attenuated organisms. I told you that they are very active, right? Attenuated organisms means you decrease or weaken those organisms, and you can use as a vaccine. For example, you see, attenuated means the pathogenicity is removed, but it is able to grow as well as multiply, right? So rota vaccine from rotavirus area. Here, what is happening? Rotavirus is attenuated so that it became weak, right? Once it became weak, it can induce the immune response. There is no pathogenicity, but it can induce the immune response. Whenever the same person is attacked with the same microbe, then that individual is protected from that real infection, because it is already exposed and he had he had immunity for particular rotavirus. Here you see killed organism, polio vaccine. You see polio vaccine or rabies vaccine. Here these microbes are completely killed and they are neutralized. When they are neutralized, when that neutralized virus injected into the humans, they develop immunity. Right? Yes or no? So I am telling you that fully active we can use, and we can modify the organism by weakening, and pathogen is removed, and that can be used as a vaccine. And here, when it is not possible, you can kill them completely, and still you can use them as a vaccine. Right? So I am trying to tell you all that. You know, you can use when you have a technology can modify the biological material to produce a useful product. So here the requirement is you need a living organism completely. And here we are weakening that, and here we are killing. So by these three ways, we are producing more valuable products out of it, right? Use part of the organism. Here, in the first three examples, we use the whole organism, but in the fourth one, you can see here use the part of organism. We are not taking the entire organism. You see, hepatitis B, right? So the vaccine is composed of protein L and MS that resides in the surface of the virus. When you isolate these proteins from the virus, and if you then we can make the vaccine. We can make the vaccine. Production of elite or endangered plants. We are here. What we are trying to tell, if you have a plant, the plant is endangered or very, very important. So using the tissue culture, we can produce those plants in billions, right? We are amplifying the number. We are amplifying those plants in a in vitro condition. Then later on they can be taken to the real soils and so on, right? So we are producing more plants from millions of plants from one or two plants maximum, right? That is through the tissue culture. Modified organism. Here we are modifying the organism through the genetic modification. As I told you that bacterial gene. You see, bacterial gene CRY is integrated into the cotton genome. And what you are getting? BT cotton plants. Produce insecticide and are resistant to the insects. That's a bollworm attack. You see here living organism, attenuated organism, killed organism, part of organism. We are producing more copies of it. Now we are modifying the genome of a particular organism to produce whatever character you want. Here we want a insecticide character in the plant by integrating the bacterial gene. This is also possible through the biotechnology. Stem cells, as you all know that stem cells can convert into any type of cell, right? So whenever we need a particular cell, to refill them, if you don't find a good donor or a matching donor, your own cell can become as resources for required cells. You see? Cell can develop into different cell types, like muscle cell to brain cell, right? Proteins. Here, all of them are organisms. Here we are producing more copy. Here genome is modified. Here one cell is converted to other cell, right? So for example, you want liver cell. If you have a umbilical cord, if you store it, from that umbilical cord stem cells, you can convert it to whatever cells you want, right? And proteins. And we are coming from the whole organism to the macromolecules. Then we go to the secondary metabolite also. So you see, horse peroxidase. See, horse radish peroxidase. Right? This is used in many applications, especially diagnosis. You can produce from plants, right? And the recombinant insulin, monoclonal antibodies. You see, this Bevacizumab, this is the anti-cancer, cancer therapy you can use. And nucleic acids, you can use them for diagnosis of disease, gene therapy, RNAi, CRISPR-Cas, or they can be used as a biosensors. Carbohydrates, you all know that carbohydrates are staple food, but beyond it, you can get biofuels from the carbohydrates. Conversion of starch and lignocellulose to the biofuels such as bioethanol or biodiesel. Lipids, based vectors. Whenever you want to, transport the drugs, you can develop liposomes by using the liposomes for transport of DNA, drugs, and diagnosis of the diseases. And the secondary metabolites, like you see, secondary metabolite. Penicillin is a secondary metabolite. That's the first antibiotic, right? So, which is isolated from the fungi. That the penicillin, first antibiotic, almost it saves the millions of lives, right? So from first to 12, you say these are the living organism completely, and here we have modified the weakened the organism. Here we killed the organism. Here we used some part of the organism. Here we made the many plants or from the limited resources. Here we modified the organism. We converted the one cell to other cell, and we got a lot of proteins from we express the proteins, and here nucleic acids are used for the disease diagnosis or replacement of a defective gene through the gene therapy. Or here carbohydrates are converted to the biofuels. Lipids are used as a liposomes, and secondary metabolites are most reliable antibiotics as of now. So these are the different examples or applications of biotechnology. Same thing, here you can see pictorial. These are the lactobacillus to convert the milk into the yogurt. The yeast to convert the wine, and this is the hepatitis virus, and this is the polio vaccine virus, and this is the this is the polio, and this is the hepatitis, and this is the plant tissue culture. You can get the more plants from the limited, small amount, and this is a BT cotton, and you can see stem cells converted to the many types of cells, and here you see first time Genentech in 1981-82 they used the human gene and expressed in the E. coli to produce the active insulin. Before that, they used to get the insulin from the pig, pancreas, right? And monoclonal antibodies, and the DNA can be used to, diagnose the diseases or gene therapy, and the lipids are used for the transport of drugs and vector molecules, and the carbohydrates can be used as resources for the conversion of biofuels. And you see secondary metabolites are the most reliable. And this is all about biotechnology definition and its application. Hope you understand my lecture. If at all if you like it, try to subscribe my channel. That's Dr. PREM-PRIMER Biotech Lectures for many updates. Thank you all. Bye bye.